we're going to Google regenerative medicine, and we're going to click on the images. First things first, 80% of your immune system is being taxed and overused for digital data relay intra-body. Second, they are screwing with the human electrical homeostasis to do all of this. That's why they have to put back special instructions in the cells, ripping apart, unzipping the DNA, screwing with that endlessly to ensure that more than just the gene and RNA therapy are clicking around. Here, you have organoids and organelles that reproduce all on their own. Organoids and organelles are programmable, reproducing xenobots. We learned about xenobots in episode 58. And over here, software-defined metamaterials, tissue scaffolding. So you see, they are very busy. Doing what? Not wanting to die. They also want to upload themselves to the metaverse in a very permanent way. They could have healed people back in 1995. They have had this whole system sitting here since 1995. And what have they been doing with it? They have been using it for themselves. It's very obvious. We're going to go over here and read Tissue Engineering and Regenerative Medicine. There is a tremendous need for new strategies to promote the regeneration of musculoskeletal, skin, vascular, and dental/oral/craniofacial tissues. Due to the large number of patients suffering from disease or trauma to these tissues, we are taking a variety of approaches to meet this need, with many revolving around the use of biomaterials that either serve as carriers for morphogenes or cells that can participate in regeneration, and others exploiting mechanical cues. These materials can typically be introduced into the body using minimal invasive techniques, for example, injection, and regulate in time and space the presentation of molecular cues and subsequent recruitment of host cells that orchestrate regeneration. Here is Figure 1. Tissue regeneration can be improved by using biomaterials to exploit cell biology. Biomaterial-assisted cell delivery. To address the current limitation of cell transplantation in rebuilding tissues, for example, large-scale cell death, we have been designing biomaterial carriers that maintain cell viability during transplantation, while subsequently stabilizing the cells, or mobilizing to disperse or engraft through a large tissue volume in vivo. Additionally, we are exploiting our expertise in drug delivery to create new networks of blood vessels within regenerating tissues and reestablish innervation as capillary networks are built. Here we're looking at Figure 2. Biomaterial-assisted cell delivery. Cells harvested for therapeutic delivery can be expanded ex vivo and modified as needed before being precisely delivered using biomaterials. Biomedical niches for host cell modulation. It is clear that cells associated with both innate and adaptive immunity play key roles following tissue damage, and control over their numbers and functional states may allow one to promote regeneration. We are developing biomaterial systems to recruit host cells and modulate them to promote regeneration. For example, we are investigating how biomaterials can be leveraged to regulate the frequency and polarization state of macrophages at a site of tissue damage, and are exploring how this impacts the regeneration of skin and muscle. The role of various T-cell populations in angiogenesis and regeneration are similarly being explored using biomaterials that regulate the number and type of T cells, and the impact of antigen-specific immune responses and these processes are being explored. Here we're looking at Figure 3, biomaterial niches for host cell modulation. Biomaterial niches are used to modulate cellular behavior without ex vivo manipulation.
So for everybody who didn't want to be tested on and targeted, too bad. If you share phenotype or DNA with one of these billionaires, well, they are much more interested in that, because they will pull those cells right out of you and into themselves. That's why everything is wireless.
Let's go over here to the National Library of Medicine, and we're going to read Harnessing the Power of Artificial Intelligence for Human Living Organoid Research. As a powerful paradigm, artificial intelligence is rapidly impacting every aspect of our day-to-day life and scientific research through interdisciplinary transformations. Living human organoids have a great potential for in vitro, reshaping many aspects of in vivo, true human organs, including organ development, disease occurrence, and drug responses. To date, AI has driven the revolutionary advances of human organoids and life science, precision medicine, and pharmaceutical science in an unprecedented way. Herein, we provide a forward-looking review, the frontiers of human living organoids, covering the engineering construction strategies and multidisciplinary technologies for developing living human organoids, highlighting the cutting-edge achievements and the prospective applications of AI in living human organoids, particularly in biological study, disease occurrence, disease diagnosis, and prediction, and drug screening and preclinical assays. Moreover, we shed light on the new research trends harnessing the power of AI for living human organoid research in the context of multidisciplinary technologies. The aim of this paper is to motivate researchers to explore organ function throughout the human life cycle, narrow the gap between in vitro microphysiological models and the real human body, accurately predict human-related responses to external stimuli, like cues and drugs, accelerate the preclinical to clinical transformation, and ultimately enhance the health and well-being of patients. The key words here are artificial intelligence, human living organoids, organoid intelligence, in vitro miniature organ models, multidisciplinary techniques, preclinical to clinical transformation. AI-enabled living human organoids in biomedicine. Here we have disease modeling, diagnosis, and prediction with biosensors and organ chip. Here is optimization of culture systems, live imaging techniques, droplet microfluidic. On this side, we have data mining and analysis, multi-omics, 3D bioprinting. And here is drug testing and disease treatment. Since the end of 2022, the advent of an AI chatbot, ChatGPT, developed by OpenAI, has caused a buzz around the world due to its human-like responses to text input. This technology is promoting researchers to reorient the development direction of all walks of life and driving their scales to brand new heights. Artificial intelligence, AI, also known as machine intelligence, is a simulation model that provides targeted services for specific scenarios by highlighting reproducing complex human behaviors such as feeling, memory, generalization, reasoning, creating, deciding, copying, and so on. The emergence of big data has promoted the training of AI algorithms on large-scale datasets, thus improving the performance of tasks. AI can be trained on large amounts of data to learn from experience, decouple, and apply causality, make predictions, optimize strategies, classify objects, adapt to new situations, and perform complex tasks. It contains machine learning, natural language processing, language synthesis, computer vision, and robotics, etc. It is noteworthy that machine learning, as a cutting edge technology in the field of AI, has been widely applied to biomedical research, disease discovery, and drug screening in preclinical assays and clinical applications. For instance, Hamamoto's team was able to accurately predict integrated survival subtypes by training the reverse phase protein array and distinguish between high risk and low risk patients. Gulshan created an algorithm by applying deep learning for automated detection of diabetic retinopathy and diabetic macular edema in retinal fundus photographs. In the face of the 2019 pandemic, deep learning could effectively distinguish from normal versus abnormal chest radiographs. Advances in stem cells, microfabrication, and biomaterials have given rise to novel, in vitro, miniature organ models, living human organoids. In general, living human organoids refer to 3D living cell structure models originating from human primary tissues, or stem cells, which present abundant cell types, genotypes, histological features, and functions similar to human real organs, especially human tissue-derived organoids. Currently, various living human organoids like brain, retina, lung, intestine, tumor, and placenta, etc., have been established to explore the effects of matrix materials, for example, types, stiffness, and degradability, and signaling factors like BMP and WNT on stem cell regulation and organoid development. For instance, Lutov's team showed that high matrix stiffness significantly enhanced intestinal stem cell proliferation through a yes-associated protein-dependent mechanism, whereas a soft matrix and laminin-based adhesion were required for stem cell differentiation and organoid generation. Let's scroll down. Of particular note, the FDA Modernization Act 2.0, approved in December 2022, eliminates the age-old requirement for animal testing of all drugs and will no longer require animal testing before entering clinical trials. This release is undoubtedly a high endorsement of organoid technology, and it has also set a wave of research on organoids in academia and enterprises. Recently, organoid technology ushered an important milestone in the field of preclinical research and drug development. For the first time, the US FDA approved a drug to apply for a new indication to enter clinical trials based solely on the preclinical efficacy data obtained in the organoid chip research, combined with existing safety data. This is the first time the FDA has included organ on a chip and microphysiological systems in the Act as a separate evaluation system for non-clinical trials of drugs. This breakthrough provides a new direction for the search and development of rare disease drugs, and also provides a new idea for drug research and development enterprises to apply for drug clinical trials and new indications. Therefore, living human organoids provide valuable and effective models for drug screening and discovery to identify candidate treatments to tackle rare and emerging diseases. Yeah, the same diseases that they created. Therefore, we can't help but think about how AI technology can accelerate the development of living human organoids in the fundamental research and clinical therapy in biomedicine. In the context of technological innovation, researchers at Johns Hopkins University in the United States have creatively conceived a technology similar to human organs, organoid intelligence, brain organoids as hardware to develop biocomputing. In 2023, this team envisioned computers running on human brain organoids and announced related research plans. In the rapid development of the information age, harnessing the power of AI for living human organoid research combined with gene editing, imaging analysis, and multi-omics sequencing could aid in the organoid system improvement, clinical decision making, and clinical trial performance analysis. AI can predict the differentiation pathway of stem cells and further production and development of living human organoids, and ameliorate the living human organoid culture environment, attempting to build more sophisticated and authentic in vitro physiological models in a highly human relevant manner. There is lots and lots of information in here. I will leave the link in the description for those who want to keep reading.